Recording in progress. A warm good morning to each one of you. May I now kindly request our dignitaries to please come on to the dais. Dr. R.S. Kumar, President ISPGR and Chairman TAS, so kindly come over to the dais. Dr. Sanjay Kumar, Chairman ASRB.
Dr. R.K. Tyagi, Vice President ISPGR, and Dr. Anuradha Agarwal, General Secretary ISPGR. On behalf of the Indian Society of Plant Genetic Resources, it's a great pleasure to welcome each one of you to this special occasion. We had a slight delay due to the travel delays caused by some of our participants who are reaching from outside Delhi. Thank you very much for your cooperation and patience. At the outset, we are gathered today for the prestigious Dr. D.S. Athwal Memorial Lecture, fourth in the series, and followed by the ISPGR Awards Function, honoring excellence and outstanding contributions in the field of plant genetic resources. May I request Dr. Anuradha Agarwal, General Secretary ISPGR, to kindly welcome our dignitaries with potted plants. To Dr. Sanjay Kumar, Chairman ASRB. Dr. R.K. Tyagi, Vice President ISPGR. I now request Dr. Anuradha Agarwal to kindly deliver the formal welcome address.
Thank you, Dr. Sherry. Sabhi ko namaskar, a very good morning, and a very warm greetings to all the dignitaries on the dais, off the dais, and all the guests who have assembled in today's function with such abundance. As we have the opportunity to present here and to recognize the contributions of the various talent-worthy researchers towards the management of plant genetic resources. Padma Bhushan awardee, Dr. R.S. Paroda, President ISPGR and Chairman TAS. Thank you, sir, for not only presiding the occasion today, but also guiding the way for the entire activities of the society and, in fact, nation-level PGR management. It's a real pleasure to have your guidance and support constantly. A very warm welcome to our chief guest and speaker of the day, Dr. Sanjay Kumar, Chairman ASRB, a very renowned scientist, and His Excellence we will be getting to know in the lecture, the fourth Dr. D.S. Athwal Memorial Lecture. So I'm sure your gracious presence will be a great source of encouragement to the whole PGR family. And we look forward to your lecture today on a very important topic on bioresources to bioeconomy, building value chains for a sustainable bioeconomy. I warmly welcome all the dignitaries of the dais, starting with Dr. R.B. Singh, the eminent plant breeder and policy maker in the field of agriculture that we have in the country today. A very warm welcome to Dr. A.K. Singh, former Director IRI and Secretary of NAS. And sir, thank you for your support in giving today's facilities very generously. A very, very warm welcome to Dr. S.K. Vasan, World Food Laureate and an eminent scientist and a very, very generous benefactor of ISPGR. One of the awards that we are giving today has been sponsored by him by not only giving the core money, but also he gives us additional funds every year to support the award. A big round of applause for him. Welcome Dr. Bhattacharya, Director of NRC Plant Biotechnology. Special welcome to all the important guests of the day, the awardees. Dr. Bera, Dr. Vinay Bhadwal, Dr. Kuldeep Singh, Dr. Kaushik, Dr. Shambir Singh, Dr. Shilpa, Dr. Sharma, and I hope I have not missed anybody. Very special welcome to the entire team of ASRB, which has come in good numbers, starting with Madam Garima, the Secretary. And I welcome all the heads of division from both NBPGR and IRI and other neighboring institutes, and all the colleagues who have joined from the international organizations. Dr. Mahesh has chosen to sit himself in the end, and so also Dr. Yash. Thank you for joining. And a special warm welcome to all the participants who have joined online. I am unable to see their names, but each one is warmly welcome today. The Dr. Dilbag Singh Athwal Memorial Lecture was started in 2018. Okay, thank you for that. So I can welcome Dr. B.S. Dhillo, Dr. S.K. Sharma, Dr. Dubey, Dr. Dandin, Dr. V.P. Sahi, Dr. Randhawa, and a galaxy of scientists who have chosen to come online in spite of being in Delhi, a few of them. And I also see our EC member, Dr. Shivraj, and Dr. Praveen Singh, and many others who have joined. A warm welcome to each one of you. And yes, now I can see Dr. P.L. Gautam. Thank you, sir, for joining. These are all people who have contributed significantly to ISPGR particularly, and PGR in general, and we are very happy to reunite today. So I was telling you about this lecture. This was started in 2018 by ISPGR to recognize the immense contributions of the world-renowned wheat and rice breeder, Dr. D.S. Athwal. He was a geneticist and a scientist and one of the pioneers of the Green Revolution. And this lecture is to commemorate and acknowledge his role in the development of the gene bank at NBPGR, while he was the vice president of Winrock International, the implementing agency of the USAID project in India. So the excellent gene bank facility that we see in this campus in NBPGR was greatly supported by him, and that's why we chose to recognize him. So by inviting eminent scientists to deliver this biannual lecture, the series tries to foster exchange of knowledge on current scientific advances to continue Dr. Athwal's commitment to innovation, research, and education in agriculture. So with this very short welcome, and I hope I've not missed out names, but I welcome each one of you for today's event. I would say enjoy the function, and a very advanced Happy Christmas and New Year.
Thank you very much, ma'am. The Indian Society of Plant Genetic Resources is indeed privileged to be guided by the visionary leadership of Padma Bhushan Dr. R.S. Paroda, who is our president and also chairman of the Trust for Advancement of Agricultural Sciences. May I now request Paroda sir for his opening remarks.
Good morning to all of you. Our chief guest and speaker today, Dr. Sanjay Kumar, my colleagues, Dr. Tyagi, Anuradha ji, and esteemed colleagues and friends, Professor R.B. Singh, Dr. Rasal, Dr. A.K. Singh, Dr. Bhagmal, Dr. Bhattacharya, all awardees, I see, and many other distinguished guests and friends. It's an important event today for this Indian Society of Plant Genetic Resources. I recollect it was established in year 87, I suppose.
87.
Yeah,
November 87.
when I was about to leave NBPGR, and it almost completing 40 years. So at that time I was founder president and didn't realize that I would again be asked to play that role. But it's a happy event for me that we are today meeting for DS Athwal Memorial Lecture. When I was, and Anuradha has already given some brief background, when I was looking for support for Gene Bank at this campus, unfortunately there were no funds and also no land. And I had to look for support, got support from JICA, but that was limited to $10 million. And that could have only provided us the building and equipment. But our expectations were more, and so I approached the USAID, and fortunately we had two well-wishers and I would say supporters of NBPGR at that time. One was Dr. Nile Brady, who was Director General of ERI and then became Deputy Secretary of USAID. And Dr. D.S. Athwal was also at ERI. And you know Dr. D.S. Athwal, he is one of the premier wheat breeder and first breeder for pearl millet hybrids in the whole world, and was associated with Green Revolution from Punjab along with Dr. Swaminathan at that time. But I found in him a real well-wisher, and he came forward and advised me in many ways how to approach USAID and get resources. So we could get about $24 million US, including the support not only for building, for training of all the scientists from NBPGR. Almost 100% of the scientists were included in this project. And D.S. Athwal, being the Vice President of Winrock Foundation, I requested him to help us and be outsourcing agency to monitor that funding support so that quality of building is also taken care of. Equipments also come procured through him, through Winrock, and all our scientists are trained in the best labs abroad. And therefore, when this responsibility came again for being the president of this society, I thought here is one person whose contribution must be recognized. And that's how we started this DS Athwal, because memory otherwise is very short, and institutional memory very few have. And even maybe you may not find these details on the website of NBPGR. So sometimes it is essential that we recognize our own. people, and so we are happy that this award is in the name of Dr. D.S. Athwal Memorial Lecture, and we also are happy that Dr. Sanjay Kumar, the chairman of Agriculture Scientist Recruitment Board, has agreed to deliver this lecture on bioresources to bioenterprise. Bioresources conservation, collection, evaluation is one part, but to make them as a bioenterprise is another. And he had in the Palampur Institute been working in this area. So we thought, because unless they have some value and provide some economic advantage to those who conserve them, possibly it will be difficult to continue conserving these resources through farmers' efforts or communities. So I won't take long. I'm very happy that you all have accepted our invitation. Welcome you all. It's a great privilege to have you and have all those who are joining us online. So welcome them also. And I won't like to stand in between you and our chief speaker, Dr. Sanjay Kumar. So I would like to request you, Dr. Sanjay Kumar, maybe you are to be introduced first also by our Vice President, Dr. Tyagi. So Dr. Tyagi, could you please come forward? And personally, I say that we are very happy. This society has done well. Over the years, it organized the first International Agrobiodiversity Congress in 2016, and Prime Minister Modi had inaugurated it. It has become a rolling event. Second was held in Rome. Third was recently held in Kunming, China, where we had gone. And fourth one will be in Colombia. And ISPGR is finding its place internationally in all these events. And we are very proud that some of these achievements, which I think in the general body meeting, Anuradha will later on explain and narrate, but society has come a long way. Didn't have much resource. It has also resources, now more than about 1 crore, and didn't have many members, hardly 500. Now it has touched the number of 1,000. So it's growing, and hopefully it will grow further with all your support. So Dr. Tyagi, please.
The Dr. D.S. Athwal Memorial Lecture, as mentioned, was instituted by the Indian Society of Plant Genetic Resources to honor the legacy of a pioneering scientist and visionary leader whose contributions laid strong foundations to modern Indian agriculture as well as to PGR. This memorial lecture stands as a tribute to his enduring influence in these domains. The fourth Dr. D.S. Athwal Memorial Lecture will be delivered by Dr. Sanjay Kumar, Chairman of ASRB, a distinguished scientist and administrator. I now invite Dr. R.K. Tyagi, Vice President ISPGR, to formally introduce the speaker of the fourth D.S. Athwal Memorial Lecture.
Namaskar. Good morning, everyone. What a good day today that we have so many people here. Dr. Paroda is there, Dr. R.B. Singh, Padma Bhushan, Dr. R.B. Singh, Padma Bhushan, Dr. Paroda, World Food Laureate, Dr. Vasal, all veterans, Dr. Bhagmal, and the youngsters also there, some students I see. Respected Dr. Paroda, the President ISPGR, Dr. Anuradha, General Secretary, our chief guest, Dr. Sanjay Kumar. So it is a privilege for me to introduce today's speaker. And much has been said about the Dr. Athwal, what he has contributed to the nation as well as particularly to the ICR system through NBPGR Gene Bank. One thing today morning I was discussing with the chit-chatting with Dr. RB Singh. So he told me one very important thing which I'm tempted to share with you also. That when Dr. RB Singh was there in 1964, he joined PAU. And as associate professor and economic botanist, Dr. Athwal was the head of the department for that division. And he was saying that he used to teach the cytogenetics course, and Dr. Athwal used to attend his classes. So what a humility or what a humble person he was. And that's why I think he is still in memories of all of us. And we thank Dr. Paroda that who was suggested to have this annual lecture to celebrate his contributions, to keep his in our memory. Dr. Athwal, and I thank you all to join this lecture from NBPGR, from IR Institutes, and join Dr. Amrada to welcome you all. My privilege to introduce the speaker, so please allow me to read it out. Dr. Sanjay Kumar is distinguished scientist and research leader, currently serving as chairperson of the Agriculture Scientist Recruitment Board, New Delhi, under the Department of Agriculture Research and Education, Ministry of Agriculture and Farmers Welfare, New Delhi. In his national role, he oversees the recruitment and assessment of scientific talent across India's agriculture research institution, shaping the future cadre of researchers in agriculture sciences. Dr. Sanjay Kumar earned his master's degree from GB Pant University, Pantnagar, and his PhD from IARI, New Delhi. He enhanced his expertise through postdoctoral training at Texas Tech University, USA, Rothamsted Research, UK, Kansas State University, USA. In 1990, he joined the CSIR Institute of Himalayan Bioresources Technology, that is commonly known as ISBT, Palampur, as a scientist, and rising through the ranks and ultimately serving as director until his superannuation in February 2023, after more than 3 decades of service. Dr. Sanjay Kumar's research portfolio spans plant pathology, metabolic engineering, high-altitude adaptation mechanism, and bioresource utilization and sustainable bioeconomy. He is credited with the discovery of the transplantation of novel carbon fixation pathway to enhance photosynthetic efficiency, characterization of unique enzyme from high-altitude plants, and significant contribution to genomic and transcriptomics studies of Himalayan biodiversity. His work has led to numerous international patents and more than 220 scientific publications, and he has mentored almost 30 MSc and PhD students. As director of ISBT, Dr. Sanjay Kumar championed the introduction of high-value crops such as hing. In fact, he is known as the hing, very popular because of the hing he introduced in the country, and very successfully developed the protocol and the in fact in the farmers in non-traditional area also that he has developed the protocol or the cultivation practices of hing. Other crops saffron, monk fruit, cinnamon and aromatics in non-traditional regions, and promoted waste management and nutraceuticals technology benefiting farmers and local economies. He is a fellow of Indian National Science Academy, NASI, NAS, and several other scientific bodies. I will refrain to name all those bodies here for the paucity of time. His honors include the Vasvik Industrial Research Award, INSA Young Scientist Award, and multiple distinguished lectureship and leadership awards. With a relentless approach to quality science and technology development led by HBT to attain the top position in the state and be ranked among the top 10 institute of CSIR as per Science Mago ratings. Dr. Sanjay Kumar's career exemplifies scientific excellence translated into social impact, bridging fundamental research and technology transfer and agriculture development. Here is the, that's what I have to introduce him. And in fact, sir, we are very thankful that I want to tell the audience that yesterday till night he was in the QM2, and we were having some level of palpitation that whether because of the flight delays and all that, whether he will be able to reach or not. But you have made it possible, sir. Thank you so much. This extra efforts you have made to join us. Now, may I request the dignitaries to offer the flowers to Dr. Athwal. I will request Dr. Vasal also, Dr. Bhattacharya, Dr. Ashok Singh, Dr. Arbe Singh, Dr. Bhagmal. Dr. Kuldeep Singh also has the legacy of Dr. Athwal being from PAU, so I will request him also kindly.
So very good morning, respected Dr. Paroda sir, President ISPGR and the Chairman of TAS, Dr. R.K. Tyagi ji, Dr. Anuradha Agrawal ji, Sherry, and in the hall we have Honorable Professor Tyagi ji, Dr. Vasal sahab, Dr. R.B. Singh ji, Dr. A.K. Singh sahab, my old good friend. Then we had Dr. Kuldeep Singh. We worked together for several years and we'll come to that also. Dr. Bhagmal sahab. We have Dr. Vinay Bhardwaj ji, my old friend from Palampur, Ram Kumar. We also have in the hall Tapan Mandal, again legacy from Palampur, and all the friends from ASRB, all the delegates, all the fellows, all the awardees. First of all, indeed it's a great honor, and I would say that it's a humble privilege to be part of this great day. Thank you so much, sir, that you considered me for this very important lecture. Particularly a person like Dr. Athwal, who is first of its own kind. What he did, how he utilized bioresources and made it that everybody today, I think if we talk of wheat, and his names come the first, although green revolution we can attribute to somebody, but when it comes to wheat revolution, I am sure that everybody would remember Dr. Athwal, the way he introduced all the genes that you all are aware of. He did it first for the first time, and today happens to be the 19th December, and in 1901 first Nobel Prize was also given on this day. So I think best way to pay tribute to a person like Dr. Athwal could not have been better than the day of today. So thank you so much, sir, and you gave me this opportunity to me for this honor. I would say that it's a great honor for me. Sir, I'll try to give you some background of what I did at Palampur. Therefore, when I say and when I discuss my work, I would like to acknowledge all the work which was done at IHBT, so all the staff there, and also my several projects, they were funded by CSIR, ICR, DBT, to my acknowledgement to all of them for this work. Sir, I thought I will try to speak something how we can generate economy out of the bioresources in a sustainable manner. And there can be at least 5 different ways from where we can derive economy using plant-based bioresources, whether it is for food or feed, whether it is for bioenergy, whether it is for bio-based materials such as fibers, or even for biopharmaceuticals, whether you talk of medicines or you talk of aromas, everything basically you are dependent on plants. Similarly, in animal resources also, you can generate these 4 or 5 different categories where economical generation is very, very evident. So I am not listing all the things what we can generate from animal sources, but I think animal resources are equally important. And therefore, when I was trying to go through various issues, I thought that Professor Parodaji can be considered as a global champion if you talk of genetic resources, while because he thought, I think if you talk of architect of genetic resources in India, you can't imagine any other name except for him. Therefore, animal slide I purposely did not put it. I thought I will like to give acknowledgement to Professor Parodaji for that purpose, because you see all the genetic animal, fish, all the genetic resources is his contribution in India, what we are looking for. And therefore, there is enormous opportunity to generate economy out of that. If you see today's bioeconomy potential of India, we started with the $10 billion in 2014, and today we have around 167 billions. It was 24 data, which is 165 billion, but by 24 we had 167 billion, and right now we are around to 4.25% of our GDP. And this is phenomenal. And by 2030, we expect to be $300 billion. And here, at least 4 different sectors can contribute very significantly, whether it is the area of biofuels, enzymes, bioplastics, diagnostic or vaccines. And in bioagriculture, I think precision agriculture is going to take place very, very significantly. And certainly, IT sector is coming up. And when we are integrating with biotechnology, it is going to play some important role in this area as well. Now, I wanted to, is it not working? Can you? Okay. So when I wanted to mention that using bioresources, you want to develop economy, at least 5 important parts are important. Unless we have enough raw material, we have technology to develop products, we have market linkages, and then its availability to consumers, and last and most important thing is the responsive support. Most of the time, several of our labs are happy in giving technologies, but when it comes to hand-holding and support, after sales service, what they call it, there we need to pay a lot of attention. And as an institution also, our sale support is very, very important for the technology. And I'll give you now few examples how we try to develop economy using the bioresources. I'll give you few examples. Now, I'm not giving you whole data of the nation that we are mega diverse country and we have all sorts of resources, but I'll give you some examples. Using some unique resources, how we converted them into the economic potential and industrial development and also taking it to the market. Most of my work was limited to high altitude. It's around 15,000 feet and above. That is the area of work I used to spend months together in those high altitude. And this picture what I am showing here, it is of Kunzam Pass. It's 1500 feet in Spiti, 15,000 feet in Spiti Valley. And what you see here is a plant. This is the most dominant significant species at high altitude. This is known as potentialized species. And beauty of this plant is it's almost green, lush green plant. Hardly you see any water. During daytime temperature will be somewhere 25 to 30 degrees Celsius. Nighttime temperatures will be around minus 20 degrees Celsius during growing season. And still this plant survives. And we thought this plant be having something very unique protein which can be utilized for some industrial purpose. So we tried to analyze this plant. I will not go into more detail, but when we were trying to systematically analyze, what we found that it has got some very interesting enzyme known as superoxide dismutase, which takes care of superoxide radical. And you know, in our body also superoxide radical is produced all the time and it leads to aging and all that phenomena. And we thought it can be very useful even from human purpose. And when we try to see this enzyme here, this enzyme was functional right up to minus degrees Celsius. You go to up to minus 20, this enzyme was functional. So we developed special assays to assay it at sub-zero temperature. You heat it and this enzyme was functional. You bring it to normal temperature after autoclaving, you will see it retains at least 70% of its activity. So we thought we have something very unique because such activities were not reported till now. And then we tried to do the structure analysis. So we found out the structure of this enzyme. So we went to ICGB because we did not have the protein crystal structure facility. So we went to ICGB. There we tried to find out the structure. And once we found out the structure, we realized that it has got some very interesting phenomena, like some unique amino acid it had. And we thought if you change those amino acid and try to make it, can we make it more thermostable or not? That was our question. So we did 8 and 9 different mutations, and after that one mutation did wonderfully well. And the scenario was, even if you autoclave it 3 times, enzyme does not lose its activity. It is stable at room temperature. You don't need a refrigerator. So we thought that now it has got something which we can take it to the industry. So we developed the cultivation protocol, and up to bioreactor, 3000 liter bioreactor we evaluated with lot of success. So we went to MTech, all other institution we explored, and once we were confident that its yield was phenomenal, around 350 milligram of this protein from 1 liter of culture, you can get so much of yield, and that's what is needed by the industry. Unless industry is a yield friendly, so economically it was very feasible, and we went to the level that you could crystallize these proteins. What you see, this coal-like structure, this is the crystallized protein SOD what we had. So whenever we had to transport, we would transport in this form. So right from conception to the technology level, it was totally ready. And all the steps that were involved in between, they were user friendly. Anybody can use it, even class 12 student was able to use it. So we made it so simple. And once it was ready, so we had all the patents, all the publication in very good journals, and we approached one company. They gave us very premium price for this technology. And even if you see this company even today, it says with pride that only company globally offering 100% purified sod with wide stability range. So right from bioresource to the industry, this was our first success story. An important point here is we wanted to conserve this resource. we said that we don't have to go back to the plant to take out this enzyme. So we developed E. coli based system so that there is conservation is at one hand. At the same time, technology was developed on the other hand. So that was our first successful example. At that time, company had given us around 1 crore rupees, and then other royalty items, those were there. But we kept everything with the institute. None of us took even a single pie out of that technology transfer. We said it should be for the industrial purpose and not for personal purpose. So everything is for the institutional advantage and not for personal advantage. So this was our first successful case that using bioresource, how we went to the industry. Second, I'll tell you one very interesting example. You know, we all are aware, when we all are plant scientists, we understand C3 plants, C4 plants. When I was again working at high altitude around 15,000 feet, you know, what our first observation was, we had a lot of difficulty in walking around. And you see that people say that people at hilly areas, they have red cheeks, and they said they are very healthy. Actually, that is an adaptation, because oxygen partial pressure is so less that their hemoglobin content has to go up to cope up with for that scenario. And therefore their cheeks are red because hemoglobin content is high. So we said, how plant adapts itself? That was our second question. When human can have hemoglobin, what plant has? Is there any new mechanism for CO2 fixation? So we here adopted a classical approach of Kelvin cycle, how we had traced the pathway of carbon fixation. So we used to do the same experiment at high altitude. And very interestingly, we found that at high altitude, plant had a very unique mechanism. You know, photorespiration used to be pretty high, but what plant would do, whenever the nitrogen, you know that in photorespiration ammonia is evolved and carbon dioxide is evolved, plant would refix those carbon and nitrogen. And to fix that nitrogen, we all know that you need a carbon. And for that, plant use PEP carboxylase, which is normally a C4 enzyme. So we found a very unique way that plant has a very nice way of refixing the photorespiratory losses of carbon and nitrogen. While whole world was trying to convert C3 plant into C4 plant, we had some very simple way that you refix the CO2 and nitrogen which is emitted during photorespiration. So we try to publish it in very good journals and they would return it. So I happened to be at Ratham State and I was talking to Lawler. I said, sir, what is the reason why our paper is not getting accepted? You know what he told me? He said, in your paper, looks like he was the reviewer. He said, in your paper, you write that at 15,000 feet, you are calibrating your IRGA, you are doing this, you are doing that. How it is possible at that height? Then I explained him all the methodology. Then he said, you give me your paper and send it to photosynthesis research, and within 2 weeks, this paper was accepted, right? And I acknowledged him in my publication also. So sometimes Western cannot believe that we can do such work. So unless he interviewed me, this paper was not published. Anyway, so first we published this paper in photosynthesis research. Then we transplanted this whole pathway in Arabidopsis. It went into plant physiology. And then we transplanted in Brassica. It went into Plant Biotechnology Journal. And today we have those Brassica lines which can photorespiratorily very advanced. They behave like our C4 plants. And I took help of NIPV also in collecting some of the seeds and trying to see that this is possible or not. And he was, Dr. Bhattacharya was kind enough to give us those seeds. And I think that paper will be coming soon now. But this was again, once we published this, then MIT picked up this work and they included in their teaching that at altitude can alter the photosynthesis. And that was a great satisfaction for us. Then in some books, this pathway went on. And very recently, you can see this recommendation in faculty opinions in 2018. They picked up our paper and they said that it is a very new finding. And he said that it's a new way that you can enhance the photorespiratory process. And once your work gets recognized internationally, I think nothing better than that. So this was our second successful story. Although I understand there are certain limitations that we can't release our GM crops at this time. But at least now we know that which QTL we can target if we have to take care of photo respiration even in C3 plant. So at least one that clue we could very clearly establish. So this was our second finding from our high altitude region. Apart from that, we thought that once we work at high altitude area, we have a lot of opportunity for synthetic biology. So our lab was the first in India at least to start and understanding why tea makes, for example, tea, why picoriza makes picrosides, why podophyllum is synthesized podophyllatoxin, why stevia synthesizes steviosides, why arnevia synthesizes chiconin. So we try to work out the molecular basis and understanding why certain plants synthesize certain molecules. So we worked out the complete pathway and regulatory elements, and later we thought that why can't we develop a system, because some of these plants takes around 5 years in the field to synthesize this molecule. We said, can we synthesize it economically viably in the lab, some of these molecules? And I am happy to share you that we succeeded. We succeeded well, at least through organ culture. Like for example, we did with valerian, we did with picoriza, we did with arnevia. You know, arnevia is ratanjot, picoriza makes the kutkosides. You know, even if liver is damaged by 50%, this is such a nice molecule. It can rejuvenate the liver molecules. You know, picoriza curva, valerian. If somebody is having sleep problem, just have some roots near your bed pillow, and you will get a very nice sleep. So I worked on such a nice plants. And roots, you know, I can guarantee you, if somebody is having difficulty in good, you know, is having some, you know, there is a problem, he is not feeling hungry, just take little bit of roots, your liver will be perfectly fine. So after typhoid, people have problem, take 2 times 500 milligram these roots and you will be perfectly fine. So picoriza is such a phenomenal thing. So we developed this organ culture, and today you can see you know, each flask is behaving like a plant, and what you take 5 months, in 4 months it is already. So new technologies can do this wonder, and I am sure that now some few companies have come forward to take this technology, and they are taking it forward. So these were some of the things I thought I will share with you on how high science can also lead to, people can say that basic science is only for basic science and it does not lead any product, and I always believe that if basic science is good, always some or the other product will be developed. So when I used to go to high altitude, I always used to interact with some of the farmers. I used to pass through villages, used to interact with them, and once farmers shared with us that we are not able to cultivate our regular crops. We will growing maize and wheat, and they said in the night some wild animals, boars will come, they will destroy our crop, monkey will come, they will destroy our crop. Can you suggest us something? So at CSIR level, we had a discussion that we had several crops which we could have offered. And you know, if you travel Himachal, you will find it mid-altitude, it's full of Tetratus minuta. And when we, this wild marigold, what you call. And you know, India imports the oil of wild marigold from South Africa. I said, here naturally it is growing, why people are importing? Then we realized people don't have the right variety, which has the right content of the oil, and the composition was not good. So our breeders were doing some improvement in the Tetratus minuta, that is wild marigold. So now they had the variety where the oil content was somewhere around 0.3%. Our chemical engineers, they had developed some distillation units. Market we had, because we are aware that we are all importing, we said, why don't we develop a complete value chain, right from right varieties to nurseries to cultivation practices to distillation units to extraction, quality, value addition, everything, storage and marketing. Up to that extent, we developed a mission mode project that within 2 years, if India is importing 12 tons, at least 8 tons we should be able to make it. You know, I understand. For Dr. Singh, if I say ton, he will say it has no meaning. But let me tell you, Dr. Singh sahab, for essential oils, ton is more than sufficient. For food grains, it is million ton. For essential oils, because rose oil will cost you 7 lakh rupees a liter. So it compensates that way. So it's a low volume but high value crop. So, right? So and when you are importing all this crop. So we started with this, and you believe it or not, within 2 years, you see there was news everywhere that Himachal became the biggest producer of marigold essential oil. And only thing is that we did complete value chain, right from raw material to the production to the market linkage. So people used to come and give our farmers the money in advance. So as a result, all those people who were leaving the villages, their children started coming back. Earlier they were in Baddi earning 8,000 rupees. Now they say that we can earn several lakh rupees sitting in our home. So reverse migration started taking place, and it was our very successful model. Several crores of rupees our farmers used to earn just by sale of this oil. So initially we started with the wild marigold oil, and now I am sure that institute must have taken some other oils also, like rose oil we were targeting, and some such oils. Another story I'll tell you, again, it was when we were interacting with farmers, when we happened to go to J&K, and that was the time when J&K had opened the tulip garden. We were very excited to see the tulip garden. We interacted with those people that from where you get tulips, and they said, sir, we are getting all of them from Netherlands. I said, what is so special about Netherlands? Why can't you cultivate in India? They said they get under some agreement and you can't use those bulbs for reproduction. I said, this is something very strange. So we tried to get some of the tulips which were basically free from all licensed business, and we developed complete agro technology and production technology, and at the right location. So our Lahore Spiti again become a very fantastic place for them. And once we developed that production technology, we developed the first tulip garden of Himachal Pradesh using the bulb produced in India. And that became a news. From Delhi, our LG office, he called us. He said that, can you put this tulip in at least Rashtrapati Bhavan, which is made in India? So we signed an agreement with that. And later on, we also developed the technology using precision agriculture like hydroponics and aeroponics, so that you can have these tulips whenever you need it. It is irrespective of the season. So that was the advantage of this hydroponics and aeroponics technology. So we developed that. And advantage of that, when sir this Ram Pratishtha took place, I am sure Professor Paudar sir had gone in that Ram Pratishtha, and it was a news headline that tulips offered to Lord Ram was provided by Palampur lab, and all the tulips were made in India. So that was the news headline. I was surprised, that some such things can make such a difference. Apart from the improving the livelihood of the farmers, that was a news headline, and it was a great satisfaction. And this year I could see that 9th Tulip Festival Delhi, and they say that 15,000 indigenized tulip bulbs supplied by CSIR ISPT. So apart from getting the money, at least nationally you are also getting recognized by some of these activities, and that gives you a lot of satisfaction. Leh, for example, people had nothing. That's a cold desert area. We introduced lilium there, and today you see their picture. First flight from Leh comes to Delhi, and their cargo is filled with lilium flowers. And all lilium bulb made in India, all lilium flower made in India. Otherwise, lilium bulbs we are also dependent on import in this country. So lilium, again, we indigenize the production of lilium production in India. And that's how you see in the market. Otherwise, they were never celebrating 12th February. Now at least they are able to celebrate 12th February. We have some flowers in that place. But whenever we are doing all these things, we try to diffuse our technology in the people's hand. And for that, MSME came to our rescue. MSME used to fund us, and through MSME we developed several centers whereby all our floriculturists, they were now entrepreneurs. They were doing packaging, transporting, everything they were doing, and MSME came to be very, very useful for that purpose. And MSME we actually deployed at several other places. apart from that, now I'll go to some other part. So when we were working with tulip at high altitude, we realized that whole cold desert area is just lying vacant, and people were using either for cauliflower or peas. And then we asked the question, what people in other country with similar geography do. So when we try to analyze Afghanistan, Uzbekistan, we realize they cultivate hing. And India is importing at least 1500 tons of hing every year from these countries. We don't have the plant of hing. So at that time, Dr. Kuldeep Singh Saab came to our rescue. So we were writing to various agencies, around 25,000 letters we wrote to various places. After 3 years of hard work, so one person agreed that we will give you 100 grams of seed or some grams of seed. I think it was more than what was actually permitted. So we requested Dr. Singh how to help it. At that time he was heading ISPGR, and he said, don't worry, you try to get it under different accessions. So we made different accessions, and he helped us a lot in getting these hing seeds into this country because it was nationally important. But then he did all the work what was needed to introduce this hing, whether it was all the legal issues and other issues. He took care, quarantine, etc. And finally, on 15th October 2020, which was the birth anniversary of Abdul Kalam ji, it was peak COVID season, but we said it today is Kalam ji's anniversary, we should try to introduce this hing on this very pious day. We did it, and when we were coming in the evening after doing this plantation, silently we went there. Nobody was aware that we had gone there. In the morning, early morning, 5 o'clock we started, and COVID was unheard in Lahore Spiti. They were not even knowing what COVID is all about. We did all the plantation. When we came at 8 o'clock, suddenly we opened by chance we opened our radio, and radio said, आज देश में पहला हिंग का पौधा आरोपित किया गया. And it was तो my driver came and said, सर ये हम लोग की खबर है क्या? पीछे हमारे 2-3 लोग और थे, कहते सर ये खबर अपनी है या किसकी है? Because none of us were aware that somebody has given this news. It seems somebody disseminated that news to the radio people, and this news became viral. So I shared with our DG sahab, Dr. Mande ji was our DG. He tweeted it, and suddenly Smriti Irani ji called. How it is possible India is consuming hing for centuries and nobody is growing hing? Is it a true news or is it a false news? Now Dr. Mande was worried that is it really true or false? So Dr. Mahapatra ji, he was the DG at that time. Dr. Mahapatra confirmed. He said, no, sir, I think in India we did not have this plant, so this is probably the right news. And then it went to PMO, and one friend from South India, he wrote, up till now I was so proud that my sambar is at least indigenous, but today I realize that my sambar is also not totally indigenous. So such comments we started getting once we introduced this hing, and it was then a matter, whether it was a BBC or India or abroad or everywhere, this news was that for the first time, India, which is a major consumer of hing, did not grow the hing. So that became a very big story, but I must at this time admit that had ICR and BPGR was not there and director like Kuldeep Singh was not there, it would not have been possible. That I must admit. And therefore, in all our papers, you must have seen everywhere we always admit, write very clearly that it was NBPGR's effort. It is joint effort, I would say, that we could introduce this hing. Another I wanted to share, I always work on those crops on which India was dependent on outside country. Another was saffron or kesar. And from Kashmir, that in Pempo region, around 3,000 hectares under saffron cultivation, and around 9 tons is produced, and we consume around 100 tons, and all is again imported in this country, and that 3 to 5 lakh rupees is very expensive spice. And we said, and everybody said that, kesar Kashmir se bahar nahi ja sakta. वो तो उगेगा ही नहीं कश्मीर से बाहर केसर. हमने कहा भाई बड़ी अजीब बात है. Then you will be surprised places like Palampur was found very suitable for production of bulbs of saffron. And we made it as a bulb production center. Tissue culture, plus take it to the, and you know how I went? I went to a person who used to grow onion. I said, can you grow something like onion in your field? And he did it with all success. So, I give this credit to that onion farmer also, because people don't believe that, saffron can be grown in Palampur, but onion farmer did not know. So sometimes if people don't know that it is not possible, then only they can do it. So then now you can see now more than 20 blocks in Himachal Pradesh, they are cultivating saffron. So saffron came out of Kashmir, and I am sure that after a few years, again it will be a story that Himachal is a producer of saffron. But at least it started there. Another story, we used to go to northeastern region, and there people were involved in cultivation of some noxious things, and CM was very unhappy. So I had a discussion with their CM and some of the MPs, and we said, why can't we start cultivating apples there, because apple is all coming from Burma and other regions. So we started, and by that time we had tissue culture labs which were making the root stocks of apple. We already had some apples which were low chilling varieties, and we realized that Northeast is suitable for that purpose. So with the political will and our support, for the first time we introduced apples in Manipur, apple in Mizoram, and you can see their chief minister is honoring all the farmers who started this apple cultivation. He was so happy. He said that earlier people were working with opium and such things. Now they have some way to improve their livelihood. They are selling 200 rupees a kg apple there. And Honorable Prime Minister then said that my people from Northeast went to Himachal to learn apple cultivation. So sometimes if you have the right technology, but in the process, all our tissue culture labs, they were empowered. In the process, all our farmers who were cultivating this low chilling variety, they were empowered. So we hired some people who started making this graft. So initially, more than 1 lakh plantation we did in Northeast, and it made an impact. So these were some of the things in terms of new crop introduction. Then we thought, we should do beyond this crop introduction also. For example, we all are aware India loses approximately 30% in post-harvest losses. We were limited to jams and jellies. We thought, why don't we introduce some new technologies, like for example lyophilization. But machine will cost you 2 crore rupees. So how you can put in Gram Panchayat? So we had one lab, CSIO. We discussed with them. They made this machine for 50,000 rupees. Now we can put it in Gram Panchayat. So in few Gram Panchayat we tried with success. Like all the local fruits, they would do lyophilization with that. And like that, we used to do some invention and innovation in that area. Another successful story is about stevia. Everybody is, people are cautious about not taking sugar because of diabetes and for several other reasons. Some say that my weight goes up if we eat sugar. Stevia, it's 203 times sweeter than sugar. So we developed the right variety which has higher riboside content, so that the bitterness is less. Otherwise in stevia has some aftertaste. So to reduce that, we developed the right variety. Then we developed the processing technology, and you can see this year in the beginning, one company, it opened a complete unit for stevia processing. So right from cultivation to processing. So all farmers' livelihood was improved and industry development took place. And on the same line, again we introduced a new crop, and again ISPGR came to our rescue. For this crop, monk fruit, it has got mongroside, which is 300 times sweeter than the sugar. Right now in the whole world, only China cultivates it. In USA it is processed and distributed throughout the world. For the first time, we introduced it in India, developed its biology, developed its pollination technology, and now we have tissue culture technology because it has male and female plant different. So all those technologies are in place. and now very big company has taken our this task. So like this, we had series of such things. We start with bioresources and try to end in some or the other technology. And I'll try to, in 3 minutes, I'll finish my talk now. So likewise, we introduce again true cinnamon. I was surprised, sir. Cinnamon center of origin is Kerala, but India imports around 45,000 tons of cinnamon from Sri Lanka and some other countries. I am surprised. And what we are importing is Cassia species, which is toxic. We wanted Verum species. So again using our Calicut Institute, we introduced in Himachal Pradesh because we said that we should start doing this. And again, right from cultivation to processing, we started this. Mulati, this everybody used for kaha, but except for HAU, nobody had cultivation or even accessions of mulati. So again, we introduced in Himachal, and whenever we try to take any medicinal plant, if we say that something has got some medicinal value, so we developed the human trials to ensure that it got the right efficacy. So here I am showing that using one plant species, we made something which can repair our knees, damaged knees, and it can be very helpful. And like that, vitamin D deficiency we worked on, and with the Poshan Abhiyan of Government of India, it helped several thousands of children in improving their vitamin D content, iron content, all those things. So like that, I have series of such stories where using bioresources, we add value and take it to the industry. And as a result, if you see in the end, we had around 56 startups which we developed around various technologies using some of this. And apart from that, we developed around 83 technologies, and out of these 83, 40 technologies were given to 88 different entrepreneurs, all bioresource based. So right from bioresource identification to various technology development, all this we did. And in the end, I must say, sir, that when we listen the contribution of persons like Dr. Athwal, today if we are wheat sufficient, having pizza to burger to everything, it's all because of him. And you see how many industries are developing around one person, how he changed the life of so many people around him. I think such an inspiration and such an inspiring story of his life. And if all of us who are sitting in this hall, certainly, sir, all of you have contributed so immensely, but our youngsters, if they can learn from his life, do something that society can be proud of us. So that is one great thing, sir, we learned, and thank you so much, sir, that you gave us this opportunity. Otherwise, it would not have been possible to understand even Dr. Athwal's work to that extent. Thank you so much. Thank you so much.
Thank you very much, sir. Sir's message has aptly highlighted the importance of having innovations based on unique plant genetic resources and not stopping there, but taking it to the last mile delivery to the right stakeholders. And in the PGR continuum, of which actually each one of us are members, it is a good reminder of our collective responsibility as agriculture scientists as well. May I now request Dr. R.S. Paroda to kindly felicitate our speaker. So this is a token representing our ancient heritage of biodiversity as represented in the relics of Indus Valley civilization, which has been specially chosen for the occasion. We hope you would love it. In view of the exemplary scientific excellence as already described by Tyagi sir, the Indian Society of Plant Genetic Resources is pleased to confer the Honorary Fellow Award to Dr. Sanjay Kumar. May I again request Dr. R.S. Paroda to kindly do the honors. Continuing with the conferment of ISPGR Awards, we now have the 14th Dr. Harbhajan Singh Memorial Award for Lifetime Achievements 2023-24. ISPGR is proud to announce the recipient of this prestigious award, Dr. Kuldeep Singh, head of the ICRISAT Gene Bank and former director ICR NBPGR. Dr. Kuldeep Singh is an internationally renowned plant geneticist and breeder who has made outstanding contributions to agricultural genomics, germplasm conservation, and science-led varietal development. His career spanning over 3 decades exemplifies excellence in research, teaching, institutional leadership, and global policy management. He has begun his career at Punjab Agriculture University, Ludhiana, and has established pioneering research in rice genetics. As director of ICAR NBPGR, Dr. Singh led the modernization of the National Gene Bank, successfully mobilized over 300 crores for large-scale national missions on germplasm characterization and utilization involving more than 30 institutions. A fellow of the National Academy of Agricultural Sciences and recipient of several prestigious awards, Dr. Kuldeep Singh's contributions have profoundly strengthened agriculture science, biodiversity conservation, and food security in India and beyond. ISPGR sir is indeed honored to present this award. The next award is the 5th Dr. S.K. Wasal Award for Efficient Use of PGR 2024. It has been jointly awarded to 2 teams. First, Dr. Vinay Bharadwaj and team, including Drs. Salej Sood, Sanjeev Sharma, and Surendra Kumar Kaushik from ICR CPRI Shimla. The team of Dr. Vinay Bharadwaj and the announced members have made significant contributions in the management and use of potato genetic resources. Their efforts led to the development and registration of over 20 elite germplasm lines with NBPGR, and these include varieties resistant to late blight and several significant diseases suited for processing, rich in micronutrients, and also for table use. Collectively, these varieties now cover approximately 30% of the total 2.3 million hectare potato growing area in India, significantly contributing to national productivity, resilience, and nutritional security. The 5th Dr. S.K. Wasal Award has been jointly awarded to Dr. Sandeep Kumar Bera, Director, ICR Indian Institute of Groundnut Research, Junagadh. Dr. S.K. Bera has made pioneering contributions to groundnut improvement in India. As a sponsoring breeder, he developed and released the country's first 2 biofortified, high-yielding, high-oily groundnut varieties. He has registered 34 novel groundnut germplasm lines with ICR NBPGR. And he has also done breeding using wild Arachis species, introgressing genes into cultivated groundnut significantly contributed to enhancing national groundnut production from 10.29 million tons in 23-24 to 11.3 million tons in 24-25. The 10th B.R. Barwale Award for Excellence in PGR 2024. The recipient of this prestigious award is Dr. Ram Kumar Sharma, Chief Scientist, Biotechnology Department, CSIR Institute of Himalayan Bioresource Technology, Palampur. Dr. R.K. Sharma is a leading figure in the area of molecular genetics and genomics, contributing significantly to fundamental and translational research of commercially important plantation crops, ornaments, medicinal and aromatic plants. His R&D outcome has been prominently applied globally, including genetic improvement of tea in Sri Lanka and Kenya. His outstanding contribution for the development and commercialization of hemp stevia on pan-India basis has benefited immensely farmers and entrepreneurs and was awarded with prestigious CSIR Technology Award Certificate of Merit for Life Sciences. Hearty congratulations to Dr. Ram Kumar. Next, we have the 16th Dr. R.S. Paroda Young Scientist Award, an honor instituted by Dr. Paroda to inspire and encourage the next generation of PGR scientists. ISPGR is proud to present this award to Dr. Shilpa Parashuram. She has 10 years of experience in the collection, conservation, characterization, and utilization of pomegranate germplasm, currently senior scientist at National Research Center for Pomegranate. She facilitated the introduction of 47 exotic accessions. She's managing a field gene bank comprising 338 diverse accessions. Field explorations she has conducted in Himachal Pradesh and Uttarakhand. She has identified and characterized an early maturing commercial variety and developed 2 hybrids, and also successfully registered 5 varieties under the PPVFRA Act. Hearty congratulations, Dr. Shilpa. The next honor to be conferred is the Dr. R.K. Arora Best Paper Award 2024, published in the Society's Journal, Indian Journal of Plant Genetic Resources. The paper is Evaluation of Recombinant Inbred Lines of Arisa glaberrima and Improved Champa Masuri for Anti-Cyanosis and Tolerance Against Neela Parvata Allusions, won by the PAU team comprising of Neha Chabra, Prithinder S. Sarao, R.M. Sundaram, and Gurjit Kaur. Unfortunately, sir has not yet made it to the venue because his students are online, and due to the weather-related issue, the train is running late. Yes, but let us join in congratulating the team for the award. We now proceed to the conferment of Dr. K.L. Mehra Memorial Award for Best PGR Student 2025. Rosemary J., MSc student of PGR discipline of IARI, is the recipient. Let's join and congratulate Rosemary for this honor. We now come to the presentation of the Fellows of the Indian Society of Plant Genetic Resources for the year 2024. Let me now first call upon Dr. D.P. Semwal, Principal Scientist, ICAR National Bureau of Plant Genetic Resources. Dr. Monica Singh, Principal Scientist, ICAR National Bureau of Plant Genetic Resources. Dr. Shambir Singh, Principal Scientist, ICR Indian Institute of Maize Research, Ludhiana, Punjab. We have one more recipient who has also been unable to join us, Dr. Anitha Pedapati, Senior Scientist, ICR Indian Institute of Oil Palm Research, Pedavegi. Warmest congratulations to all the recipients, and thank you to the dignitaries for handing over the awards. We now await the President's remarks from Padma Bhushan Dr. R.S. Baroda.
I hope you will all agree that we had a very thought-provoking and very excellent presentation of Dr. D.S. Athwal Memorial Lecture by Dr. Sanjay Kumar. Thank you very much for delivering such thought-provoking and stimulating lecture, which is quite different than what we had been all the time talking about as far as genetic resource conservation is concerned. Exploration, evaluation, introduction, and then conservation. But here it is value addition and linking bio resources to bio enterprise and bioeconomics. So this is very important. Without this, possibly the whole story will be incomplete. I won't repeat what he had said, but 2-3 important messages came that we need to do research to have value addition. And without that, it will not be possible. And there you would require people who are committed to contribute and who think about society, not for themselves. This is a best example, Dr. Sanjay Kumar. We are proud of you. And from discovery to industry, what you had indicated is giving hope for young generation to make best possible use of, and that too of not only our indigenous plants but also very valuable plants from outside which we didn't have before, and hing was perhaps the best example cited. So we need to continue this effort of introducing. We introduced kiwi fruit. I recollect when I was director ISPGR, the efforts were there. Now it has become a crop in India, and production of kiwi fruit also has started coming in the market. So that shows that efforts would be required. Tulip, lilium, and all these were also mentioned, and saffron, taking from one region to another one, and apple. I would say even apple, we have a farmer, Hariman Sharma, who got Padma Shri Award, and those apples can be grown in plains, even on my own farm. I had been planting in the arid region. So the potential is there. We think that apple can only grow in the hills, but they can also now grow in the plains. So that is what is a remarkable possibility. And for all this, we would require young generation to come forward, be entrepreneur, be not job seeker but job creator, and in the process they would require institutional support, institutional support and mentorship of people like Dr. Sanjay Kumar, and hand-holding, without which that success would not be possible. And for that, we would require also eminent scientists who today got awards. It is really very encouraging that those who have been contributing significantly through their life got the fellowship. Dr. Sanjay Kumar, we are honored that you accepted our honorary fellowship. There are very few honorary fellows of this society. And Harbhajan Singh Award is the most famous award of this society. Dr. Kuldeep getting. Harbhajan Singh is our Indian Vavilov, all you know, without whose efforts the collections of genetic resources in those times when there were no roads, there were no vehicles, and there were no even tracks for people to go to remote areas. He went and collected, and he is the contributor of creating, and he was the teacher of Dr. M.S. Swaminathan for his master's degree. And through him, the NBPGR therefore could be established from the Division of Plant Introduction of IRI. So we are very happy, we are very proud that Kuldeep, you this time got this award, and congratulations to you. Congratulations to all other awardees for their excellent work. Dr. Vasal Award, Dr. Barwale Award, and then the other Young Scientist Award. They are the hope for the future because they have to take this all responsibility on their shoulders to make a difference in future. And we have come a long way, and it is a matter of great pride that in the last budget, the new gene bank, safety gene bank, based on a committee that we submitted a report, was approved, and the Prime Minister himself announced it, and efforts are now in progress to have it established in Himachal Pradesh. So that's also it will be Indian soil board, perhaps the only developing nation to have its own soil board like that we have for global collection of genetic resources. So this was a great demand for a long time. We are happy that it has, and 500 crores have been sanctioned for it. ICR is working hard, NBPGR is working hard, and efforts are now on to move ahead with the construction of the gene bank, possibly in next 1 year's time. So with this, I would not like to say much because we have exceeded our time limit by 10 minutes, but would like to once again thank you, Professor Sanjay Kumar, for your excellent talk. Maybe you give a big hand to him, please. And to all our awardees, also another big hand. And all to our today participants, give them also a big hand. Thank you very much.
We are pleased to have with us the recipient of the Dr. R.K. Arora Best Paper Award. We have Dr. Sarao. Thank you so much, sir, for taking all the effort to come over here, even with so much of weather-related issues. As we now conclude this program, it is my proud privilege to extend our sincere gratitude to Dr. Sanjay Kumar, Chairman ASRB, for delivering an enlightening Dr. D.S. Athwal Memorial Lecture. We are deeply thankful to Dr. R.S. Paroda, President ISPGR and Chairman TAS, for presiding over today's program and for his constant guidance and support. Our appreciation and gratitude to extended to all awardees for their outstanding contributions in the field of PGR, especially those who have joined us from outside Delhi, Dr. and Mrs. Kuldeep Singh, Dr. and Mrs. Bera, Dr. Vijay Bharadwaj, Dr. Shilpa, Dr. and Mrs. R.K. Sharma, and all those who were here as representatives of the award function. I also thank profusely all who have waited patiently and shown their love and passion for PGR by joining online. We have a huge array of dignitaries online, and we personally thank each one of them for their constant support, advice, and guidance, and we look forward for such future cooperation also. We also thank all our distinguished guests in the hall, including directors from sister institutes. We have Dr. S.K. Vasu, Dr. R.P. Singh. We also have Dr. A.K. Singh representing NAS, and also head of the divisions from NBPGR and also from our neighboring IARI. We also thank all the colleagues and members of the life members and annual members of the ISPGR Society for their gracious presence here, and also the executive committee members, especially Dr. R.K. Tyagi, Dr. Anuradha Agarwal, Dr. Kuldeep Tripathi, and all other councillors. We also have a special thanks for the ISPGR Award Selection Committee, comprising of Dr. S.K. Vasu, Dr. B.M. Prasanna, Dr. R.C. Agarwal, and Dr. Pratibha Brahmi, for taking all the effort of going through all the applications and making this wonderful result. And also Dr. S.K. Vasu, sir, special thanks for the generous donation of the award money. Thanks to NAS again for the support for this facility, especially Dr. A.K. Singh, Secretary NAS, Dr. Lakra, along with their staff members, Minu, Kishan, Jay Kishan, and others. Support of the staff of TAS, Dhruv, Simi, and Surjit are acknowledged. All EC members once again, and also staff of PD cell of DKMA for their constant support. And each and every participant for their patience and for the love of this particular cause of PGR. I request all of you to please now join us for lunch, and also to life members and society members to join for the GBM after this particular lunch event. Thank you once again.
Thank you, Dr. Sherry. Special thanks to Dr. Sherry for not only conducting the session, but also providing extra support for the entire event. Just a request that everybody must have lunch and then not go, but attend the GBM. We have shifted the GBM to the committee room for ease of operation. So once we have our lunch in the atrium, we will move towards the committee room 1 here on the ground floor. And I will request all our online dignitaries to rejoin us at 2:30 p.m. We'll be holding the important general body meeting. We look forward to your input in that. Thank you all and have a nice lunch.
Recording stopped.